Talk a little bit about is how do nerve impulses travel. This is going to be a little bit of dealing with how do they travel within a cell as well as in between cells. Uh, Again, we talk about a neuron getting stimulated. We're going to have a cell that's sending a message and one that's receiving the message. Once, like it says, once stimulated, a neuron's going to have to communicate information about what happened. Sensory neurons, they are going to provide information about internal and external stimuli. A lot of times they're going to be taking these back to association neurons, those inner neurons, which are then going to, in some cases, kind of work together with motor neurons or efferent neurons to send a message out to something else. But again, when we're looking at communication within here, we need to know about how do you get from the input, which is the number one here, input at the neuron, to the output of a neuron. So what is being part of that? How is it sending those signals? The other question is that space in between these ones, how do we go from one neuron to the next? We know we have probably billions or trillions of neurons, and we're then again having to know and talk about how all these are interacting with one another. So the first question there, we're going to talk about action potentials and how we're kind of dealing with those ones, either graded or action potentials. And then we know the other one here, we'll have to worry a little bit about these neurotransmitters that are moving across the synapse. So very similar to muscle cells, there is a resting potential on these nerve cells. They're not quite as negative. They have a polarization of about negative 70 millivolts. Again, I'm not super worried about the exact number of millivolts. You'll see I didn't really ask you guys that type of stuff on the test for the muscles at all. But again, these ones are about negative 70 millivolts. And again, it is due to those sodium potassium pumps and those differing kind of concentrations of those sodium potassium inside and outside the cell, as well as some other negative ions that may be on the inside or the outside of the cell. Well, we know, just like with the muscles, if we have channels for these ones, we can cause a change in that membrane potential. And if we have voltage gated channels, we can get action potentials that can travel. And we know this can be part of answering the same question we did with the muscles about how does this signal get from one area of a cell to another. Again, just to remind everybody on these ones, there is these sodium potassium pumps. So 3 sodium out for every 2 potassium in. Membrane, much like the muscles, slightly leaky to potassium, very impermeable to sodium. And these, using energy, we're going to pump these out here. This is part of what is causing that membrane potential to be different across here. We also know that we have leak channels that either are going to let some stuff leak in, or gated channels. So we have some leak channels that let certain stuff move in or out, depending on which ion we're talking about. There's also these different gated channels. So chemically gated channels are ligand gated channels. These are ones that might be normally closed, and when they get a certain ion or certain neurotransmitter binding to them, they can open up. So there's the idea of those ligand gated sodium and potassium channels. The other thing we're going to see in here, we are going to have voltage gated channels. So much like with the muscles, we're going to have voltage gated channels that are normally closed. But if that local area surrounding these ones gets to a certain threshold value, it will cause these gates to open up. So those voltage gated sodium channels that we talked about in muscle, we're going to see something again similar in the nerve cells on this. What's interesting about these sodium gated channels, they actually have 3 different states to them. Again, these are either going to a lot of times be open. Most gates usually are open or closed. We're going to see that voltage gated sodium channels actually have 3 different states to them. They have 2 different gates, an activation gate and an inactivation gate, and therefore they can kind of have 3 little stages to them here. So at resting stage, the inactivation gate is open, the activation gate is closed, but we're not letting any sodium in. When we activate this, that inactivation gate is still open, but now we open the activation gate. Might be a voltage change in this case here, and now we can get sodium ions moving through the channel. To shut this down, that activation gate stays open, but the inactivation gate is going to close for a second here, which is stopping that, and then we are going to reset this where the inactivation gate is going to be closed for a second longer. The activation gate is going to close, and then the inactivation gate can open back up, and then we're back at that pre-state ready for the next type of impulse to travel through this membrane. So when we look at a plasma membrane of a neuron, we have leak channels of both sodium and potassium in the membrane. There's a lot more potassium leak channels than sodium channels. So it's easier for potassium to move in or out, out of the cell with its concentration gradient. We definitely have the sodium potassium pumps, which are again going to be involved in maintaining this resting potential. What we're going to see when we look at this is we have what are called receptive areas of this. So this is areas where we have chemically gated channels or ligand gated channels. There's not voltage gated channels here. So a lot of, on like a multipolar neuron, this is mainly going to be those dendrites in cell body. Again, these gated channels here, they might be sodium, they might be potassium, they might be chlorine or other ions. What we're going to see with the neuron, at least a multipolar neuron, is you start getting to the axon hillock, that's where you're going to start seeing these voltage gated channels of sodium and potassium, the initial segment there. We then have conductive segments. These are ones that are down the axon, going to be, again, these voltage gated channels on these. And then finally, we get to the end, we call it the transmissive segment. This is where you have voltage-gated calcium channels. They're going to allow vesicles of a neurotransmitter to be released at that transmissive segment where it can pass it on to the next cell or effector. So, and again, this is kind of showing that idea right here. So you can see a multipolar neuron. These are all ligand-gated channels up here. Again, we don't want this to be like a muscle cell because we want this to be able to process. And one of the things here is if you have a voltage-gated channel everywhere, like a muscle cell does, once you reach threshold, it's going to travel that whole cell. What we want being able to go on up here is we want multiple signals to come into these different dendrites without an action potential happening. So if it reaches threshold at this axon hillock, that's what's going to determine whether or not we send an action potential. Once we trigger an action potential here, it's reaching threshold here, and that's going to send the message all the way down. And again, this is with that multipolar neuron. If you start thinking about bipolar neurons, I can try to grab the slide a little bit later on here, but if you think about bipolar neurons, those are the ones that had one single dendrite, one single axon. So they might have more of that conductive segment from the dendrites to the cell body, and then another conductive segment from the axon hillock down the axon. Unipolar neurons, on the other hand, they have that initial segment is going to be up at the dendrites, and then it's conductive all the way down. So there is no processing once it starts that one with like those sensory neurons. So I'll see if I have a chance to, if I have a slide a little bit later on here, or maybe the next show I'll kind of show some of this on a slide here. And again, what's important on here is we know that we have inside the cell, we have a lot more negatively charged ions and proteins and potassium. On the outside, we have negatively charged phosphate ions, negatively charged proteins. On the inside, positive ions here in terms of the potassium. And the interstitial fluid, the extracellular fluid, it's going to be much more sodium rich, and there's going to be a lot more chlorine ions, so these negative ions. And again, depending on what we have in the membrane, these ions are going to be able to transfer through here. So if we have leak channels for any of these particular ones, they can move in. If we have gated channels, they can move in based on their concentration gradients. So again, like if potassium's higher on the inside, if we open a potassium gate, it's going to move with its gradient trying to go out of the cell. There can be some issues with electrical gradients here as well, so it's going to move with the chemical as well as the electrical gradients. The main thing is if you have a positive ion that's trying to go out to an area that's much more positive, it's going to be inhibited a little bit by that electrical same charges. The electrical gradient, like I said, difference between the electrical areas and the inside and the outside of the cell. So like it says here, differences of total positive, total negative across this membrane. They're going to have an electric gradient. Most of the cells we've been talking about with the muscle and the nerve cells, they are inside is relatively more negative than the outside. And again, that's that memory potential. This is what creates that electrical difference. The chemical gradient, that is actually, is it a sodium or potassium ion? Potassium much higher on the inside, sodium much higher on the outside of the neuron, for example. When we start talking about how this stuff is going to flow, changes in the amount of difference in the electrical charge is that voltage. Generally, we're measuring this in millivolts. Again, gives us a good idea of how big of a difference there. If there's more difference, we can have a bigger change. Current is the movement of these charged particles. So the greater the movement, the greater the current, and that's what we're going to be doing order to send the signal down these ones. Anything that stops that is going to be that resistance. And again, Ohm's law, which is kind of a physics thing here, we don't get into it too much, but the idea is current is going to be a combination of that voltage divided by how much resistance there is against it. So if we have a larger voltage and less resistance, we are going to get a greater flow of these ions, or greater flow of that electricity. So potassium ends up being the really important factor when it comes to our resting membrane potential. And again, it's that electrochemical gradient that is really kind of important in this. We have a very steep concentration gradient that it's going to try to be leaving here. And if that does leave, there's a lot more negative charge stuff on the inside of the cell. We do have an electrical gradient kind of opposing that. So as we get more potassium ions out of there, it becomes more difficult for more to diffuse out. But again, at equilibrium, we have a chemical concentration gradient that's equal to the opposing electrical gradient, so we're not going to be getting movement. The resting membrane potential is about negative 90 millivolts if only potassium channels were present. It's a little bit less than that because we have those sodium gates and they're having a little bit more ions on the outside. So that typical neuron, like I said, is negative 70. If we were only looking at potassium, it'd be about negative 90. But that sodium movement brings it back a little bit. So we do have a little bit of these sodium leak channels, this stuff moving in there, but again, then we continue to pump it back out. And there's not nearly as leaky on that sense. Again, those sodium-potassium pumps, they help to establish that resting membrane potential, but it's not the only role there. The biggest role is, again, the difference across the membrane. These do pump 3 sodium out for every 2 potassium pumped in. The bigger thing here is they maintain those chemical gradients here. It's not as much the charge, but it maintains those chemical gradients. And again, neurons, this is a big part of their energy expenditure. They're doing these pumps all over these membranes, so neurons cost a lot of energy, and a big portion of that, about 2/3, is just due to these sodium-potassium pumps. So this is kind of showing you at a rest where nothing's going on here. You have the different leak channels, the gated channels, and other stuff like that. This is showing you those sodium-potassium pumps. It's going to be about a negative 70 millivolt difference inside relative to the outside. So what we'll be picking up with the next time is we're going to get into what actually happens when a signal comes to this neuron. How does it go about sending this signal? What is going on with these different potentials, what we'll call as a graded potential versus those self-propagating action potentials. So that's what we'll talk about next time.